tool use. And so I thought, here's a monkey, even though it's much smaller than Ardipithecus. Here's a quadrupedal monkey that kind of quite naturally is inclined to walk on two legs, and let's study it. What kind of gait does it adopt when it gets up on its hind limbs? So we got some capuchin monkeys in our gait lab at Stony, Brook, and this shows you a little bit of the setup. Our gait lab has a lot of similarities with human gait labs, but there are also some quite different features here. So we have multi-camera systems here that we use to film the animals. We have force transducers that tell us about the forces they exchange with the ground when they walk over it. But they walk in a tunnel here because otherwise they would be all over the place. So you cannot direct a monkey to walk this direction, walk that direction. And we have a full-time animal trainer that actually teaches them or tricks them into doing what we want them to do. And there's some peanut butter involved and other treats, to make them actually move. And so what is similar to a human gait lab is that we kind of put reflective markers on these guys, and this is a little fuzzy image from one of the videos here. And so we track with multiple cameras, we track these reflective markers and then calculate 3D coordinates of joints. And here are two force transducers, and what we often also do, depending on the question, is we attach something to these force transducers to make them more similar to the natural substrate that these animals use. So here's a quadrupedal monkey, and here's a PVC pipe that is kind of branch-like in a way. And you see the forces that the monkey generates as it moves on these branches that are attached to the force transducers. This is what some of you are more familiar with from human gait lab, where mostly ball-shaped reflective markers are used, and the cameras actually only track the 3D markers. So it's not real video, but it's the coordinates of these little balls here. That's a system that I've seen this morning in Catherine's lab. So we do real video because the animals, they wouldn't tolerate these markers. We tried that, and it takes them 5 seconds to not only take them off, but also eat them. So what we actually do is we use flat markers, and the animals also wouldn't let us shave them. So that's another problem that we don't have in people. They are quite furry beasts, and so we have to shave them and then kind of put these markers on, and they wouldn't, kind of let us just hold them. So by the time I would hold a capuchin monkey and put all the markers on, I probably would have big bite marks all over me. So we let the animals walk into this transport cage here that hooks up with their home cage, and then we have a sliding door that goes down, and then we have a monkey. And this chamber then doubles up as an anesthesia chamber. So once the monkey is in there, we hook it up to isoflurane gas, and then the monkey goes down in several minutes, and then we take it out, put a mask on it, and then kind of we do our shaving business and attach the markers. And then the monkeys, they usually wake up after only 3-4 minutes, and they are immediately interested in food. They start moving. It takes them 2-3 more minutes to kind of get coordinated, and then we collect our data. And here you see an anesthetized capuchin monkey with all these nice but flat markers on their joints. We often put a permanent marker circle around these kind of more prominent light or reflective dots, so that we can kind of consistently kind of place these markers in multiple experiments, in the same position. Yeah, and then the monkeys go into the tunnel, and I think this is the video that. Oh no, I'll show this later. We had chimpanzees in the past that were well trained, and with chimpanzees we work without a tunnel. So they would take the tunnel apart probably in no time. They are pretty strong, and they are also more cooperative. They are interested in people. They come towards us. They follow an animal trainer for treats of course. And so the cameras, what they have to learn is to stay away from the equipment. So, and that is, kind of, they have to accept that we are the alpha males. So if I stand between the camera and the equipment, I'm in alpha position, the animal will stop right there and not go further than that. And in the past we were quite successful with that, and he is one of the new kids on the block, so he's much smaller and he's still learning, lots of treats involved, and we haven't had them in the room with equipment yet because we don't trust these guys yet. So we are working with them for several months. They have learned to go into the transport chamber, and hopefully they will soon be ready for recordings. And now I have to go back. My slides are out of sequence here, and hopefully I'll be able to show you a video of the bipedal capuchin monkey. Yeah, here it comes. And you see how we make them move bipedally, just hold a bait, or kind of put a bait at eye height in front of them, and here it goes. So that's how it works. And now I want to give you a little bit of background on the mechanics of gait. And most of it goes back to Giovanni Cavagna, to the 1960s and 70s. And he has developed the models of, described the mechanical principles that drive walking and running gaits, not only in humans but also in quadrupeds. And he compared walking gaits with a rolling egg, where you kind of you vault over a fairly extended leg. The center of mass raises, and you gain potential energy, and then as the center of mass moves downward, you gain speed and with it kinetic energy, and then at the end of the step you kind of start gaining potential energy again. And this is like a rolling egg, kind of slows down and tries to get to the top, and then kind of starts rolling again. And what you find in such mechanics is an out-of-phase fluctuation of potential and kinetic energies of the center of mass. So the potential energy is highest when the center of mass is at the highest point. The kinetic energy is highest when you're on the downward slope of this arc here. So they fluctuate out of phase. And supposedly this makes walking gaits very energy efficient, because, for example, in humans we know from EMG studies that a lot of them have been done at Stony Brook, that the muscles in the hind limb for most of the stance phase are actually quite inactive. So there are major muscle groups like the gluteal muscle, the quadriceps muscles, that do not fire during stance phase. So it's a passive mechanism, as muscles are the engines in our body that eat up the energy. So we think that kind of using a pendulum-like swinging motion with an exchange of potential and kinetic energy is one of the contributors that make a walking gait quite efficient. And efficiency seems to be kind of an important feature that drives the evolution of gaits. So it saves you energy moving from point A to point B. So walking gaits have a lot in common with bouncing balls, where as you land on the kind of slightly flexed leg, it flexes more. So your potential energy, the center of mass, kind of moves down, and the kind of the tendons of these extensor muscles here, they are then stretched out. And during the second phase of stance, they rebound, they recoil, and release energy. And in such a gait, the potential and kinetic energy, they fluctuate in phase. So the body is moving fastest here at the highest phase during the aerial phase, where the center of mass is also highest. So these are the two peaks. And as you land here on the foot cuff, you slow down. At the same time, your center of mass drops down, so that's the minimum here of the potential and